So I will kick us off since there's a lot to get through today. So first of all, welcome everyone, and thank you for attending today's seminar, which will cover the cost drivers of cultivated meat. And this is really meant to be an educational resource for the community on how to think about costs for cultivated meat, where I will review the progress to date and discuss some of the remaining gaps and challenges. So I want to give a heads up to everyone that this is a pre-recorded presentation due to its length of around 90 minutes. And I wanted to ensure that all of the information was coming across clearly without a lot of sort of interludes in between. So please do feel free to grab some food or coffee while you're watching. I will be here to answer questions throughout in the chat and also at the end. And so please do drop questions in the Q&A box, not the chat box. Please also feel free to share any feedback directly with me afterwards. You'll see my information in the first slide. So things that you think I got wrong or right, that's really important for me to hear for feedback. And my role really relies a lot on triangulating data points from different individuals. So Renee, if you want to pull up the recording and we'll get started. Thanks. Welcome everyone. My name is Elliot Swartz, and I am the principal scientist specializing in cultivated meat at the Good Food Institute. And today we're going to be talking about the cost drivers of cultivated meat production. If you enjoy what you're hearing in this presentation and want to connect, feel free to follow me on Twitter, connect with me on LinkedIn, or shoot me an email if you want to follow up. So the Good Food Institute, or GFI, is a nonprofit developing the roadmap for a more sustainable, secure, and just protein supply. And we do this in programmatic work in science and technology, corporate engagement, and policy, where we're really trying to create this innovation ecosystem that is going to successfully bring alternative proteins into the marketplace. We have teams spread across several key regions around the world, and all of our work is funded entirely by philanthropy. And so if you find this presentation valuable, please do consider supporting us. Now, before we begin, I wanted to cover the purpose of this presentation is really to describe our current understanding of the major cost drivers of cultivated meat production. So I've been in this field for about 6 years now, and I think we're at a point where we can really start to define the cost drivers, as well as begin to talk about some of the progress in addressing some of them. And so what I want people to walk away with is an understanding really of how to think about this problem that is pretty complex, but can be boiled down into easier ways to sort of bucket. Now, with that said, the presentation doesn't comprehensively capture every minor cost or process consideration. For example, cell line influences are not described in too much detail here, but I think we cover really the major cost drivers. That's the intention. Now, it also should be said that we're still learning about the costs of cultivated meat production. The first companies in this field were founded about 8 or 9 years ago, and many companies are just beginning to get into their first production facilities. They're just beginning to scale up. They're just beginning to introduce products onto the marketplace. And so there's still a lot to learn about what this might look like in the future. So some cost factors today that I talk about may become more minor as new solutions emerge. And we may look back at some of the numbers in this presentation 5 years from now and realize that they were wildly wrong. And so accordingly, I'll be updating the information in this presentation on an annual basis, taking in what I think are the most important scientific studies or other data points that provide insights into these challenges. So let's start with fundamentals, really understanding what are the major drivers for cultivated meat. And we'll start by looking at this summary table from Dave Humbert's study, which is probably the most detailed and cited techno-economic analysis that has been published to date. And we'll be talking more about these numbers later on in the presentation. But what this study shows, as well as other techno-economic studies show, is really alignment into two major categories of cost drivers: the cell culture media and the infrastructure. And if we look at another study that was published, again, a few years ago by CE Delft, where they looked at the current state of production as well as future scenarios for production, you can sort of see that growth media is the current cost driver of production. And as we'll talk about, we think that there are tractable ways that growth media costs can come down quite precipitously on a relatively short timescale. And if and when that occurs, then infrastructure really becomes the longer term cost driver. So we can therefore think about cost in these two major buckets, the cell culture media and the infrastructure. And within each of these buckets, there's two separate subcategories that I think are really what should garner the most attention. So growth factors and amino acids as inputs into the cell culture media, and the buildings and the bioreactor equipment that comprise the major infrastructure for manufacturing. Now, this is not to say that other things like utilities and labor do not matter, or that there are not other media components, equipment, and consumables that contribute to cost. But the major point here is that until we address or make progress in these four major sort of subcategories, these things will continue to be smaller pieces of the overall pie. All right, so let's take a deeper dive into each of these topics, starting with the cell culture media. Now, to first sort of ground ourselves in the composition of media, we refer to the basal media as really the key nutrients that enable cells to survive in culture. And the basal media will typically contain an energy source like glucose or pyruvate, as well as amino acids, vitamins, or inorganic salts. And if we look here on the left, we can see a table of a popular basal media formulation called DMEM-F12. This contains around 50 to 52 ingredients, depending on what exact formulation you get. And it has served as a pretty useful starting point for mammalian cell cultures. But not all basal media are created equally. And so if we take a look at other media like L15, this contains far fewer ingredients, around 30 to 32. It was developed for insect cell culture, but in the context of cultivated meat, it's been used as a starting point for fish and crustacean cell cultures. Now, if you're growing cells in a basal media, you're going to have those cells survive, but they're not actually going to do much growth. And in order to get them to really grow in cell culture, you need to add in added factors. And this is a broader category of ingredients that spans things like lipids or antioxidants, hormones or growth factors, small molecules, peptides. There can also be process-related ingredients, so things like anti-foaming agents or surfactants may be included in this sort of added factor category. Now, to make a complete media, you want to take your basal media and your added factors and add them all together. And Essential 8 is an example of this that has been used in a lot of the models in the industry. It was originally developed for the growth of human stem cells, but it's useful for modeling because it really contains 8 components. So there's a basal medium of DMEM-F12 plus 7 added factors. And so you have the complete media as the basal media and the added factors. But the key point to realize is that the complete media is really going to differ by the cell type, the species, whether those cells are proliferating or differentiating. The process conditions, as well as the end product, might also influence your overall ingredient list. All right, so now that's some background on cell culture media composition. Let's talk about how we actually reduce media costs. And I think you could really boil it down into 2 major steps, where the first step is sourcing lower cost ingredients. And this is where I think, the bulk of R&D attention has gone to thus far. And when you frame it in this context, you're really optimizing for dollars per liter of media. And so you'll see this metric being used a lot throughout, scientific publications or discussion in news articles, et cetera. And what's nice about sourcing lower cost ingredients is that many of the ingredients that are used in cell culture media are really going to be used by everyone. And so by sourcing a lower cost ingredient, there's a lot of cross-industry relevance where you can help really everyone lower costs across the industry rather than just a single company. And as I'll discuss in coming slides, we think that you can have success in sourcing lower cost ingredients on a pretty short timeline. Now, what I think is often missed in the discussion of media cost reduction is that this is really only half of the story. And you can have a low cost media, but if you're using a lot of it in your process and you're using it inefficiently, then your costs are still going to be high. And so you really want to use less media overall in your production process. And by framing it this way, you're therefore switching the metric from dollars per liter of media into optimizing for the feed conversion ratio. We're thinking about it in terms of the grams of ingredients in the media that it takes to produce 1 kilogram of meat. Now, this is a slightly harder challenge because ultimately this is going to have to be tailored to each company's cells, the cell's respective metabolism, the process, as well as the products that that company might be creating. And therefore, we think that this is probably on a longer timeline to see success in this in this step. But make no mistake that success in both of these approaches is really going to be required to reach cost competitive cultivated meat production. And I'll talk a little bit about how this step is also really important and crucial to achieving sustainability goals for the industry as well. So let's dive a little bit deeper into how we're tracking success along these two major steps for cost reduction in cell culture media, where the first is really switching from pharma grade to food grade ingredients. Now, $1 per liter of media has been used as this sort of early target or benchmark for the industry. The VC firm Lever VC came out with this sort of guideline around certain metrics when they're looking at companies around the Series A stage, where they state that between $10 and $100 per liter is pretty good. $1 per liter is a longer term target. And maybe if you're talking about cents per liter, then you might be making stuff up. And you can see in other techno-economic studies that have been published that if you're able to get costs below $1 per liter, then you might start entering these cost ranges that are more competitive with conventional meats. Although in this particular study, it requires also very large scale of production, which we'll talk about later on. Now, for anyone that has really grown cells before in a lab or in graduate school, you know that most commercial stem cell media costs hundreds of dollars per liter. And so people are really perplexed by how the cultivated meat industry is going to reduce the cost of media by orders of magnitude in order to get products onto the marketplace. Like, how can this actually be possible? But I think a lot of people just don't really think about what media is. It's compositionally, it's basically Gatorade with some proteins in it, and maybe a few other added factors. It's really not that complex from a sort of nutritional standpoint. And the first person that really looked at this in detail was my colleague, Liz Specht, who published an analysis that sort of showed that you could lower media cost ingredients by sourcing food grade basal media components and making some sort of modest assumptions around scale up of some of the more expensive components in the media, like growth factors and proteins. And you can come to a conclusion that media costs can drop precipitously over 99% and also breach this dollar per liter threshold and potentially go even as low as around 25 cents per liter. Again, in this particular analysis, this essential 8 media was modeled, but the general principles sort of apply. And reading between the sort of lines here with these scenarios, you can also come to the conclusion that not only is this tractable, but it could probably happen pretty quickly. Now, anecdotally, some companies have already said that they've broken through this threshold. Some of them have begun talking about this in news articles. And I think we'll begin to see that this becomes verified in the peer-reviewed literature in the next, let's say, 6 to 18 months. So what does it mean to source a food grade ingredient? What does pharma grade and food grade mean in this context? Well, for pharma grade ingredients, we're really talking about meeting specifications at high levels of purity, as well as other specifications that are relevant to ensuring the sort of safety of use in medicine. But what a lot of people also don't realize is that not only are a lot of basal media ingredients already available at food grade, but that food grade ingredients from a purity level perspective are really not that far different from their pharma grade counterparts. And so you can see on this table in this study by scientists at the Japanese company Culture, they've basically designed the basal media formulation DMEM and sourced all of its components at food grade purification levels just through suppliers that already exist in Japan. And they've shown that you can really use this 100% food grade basal media, which they've adapted to call IMEM 2.0. to grow a variety of different cell types from different species successfully. And there are other data points from others in the industry that sort of corroborate this story, where most of these media ingredients are already available at food grade and beginning to be integrated in a lot of companies' media formulations. And a lot of people in the industry have known about this for several years. I mean, as far back as 2021, Nutraco, which is an ingredient supplier that has partnered with multiple companies in the sector, was presenting data that showed that food grade amino acids can perform similarly well compared to their pharma grade counterparts, and that almost all typical basal media ingredients are already available for use as food grade. They also showed that you can source what are called feed grade ingredients like glucose that can also perform similarly well to pharma grade counterparts. And so the story here is that, there's not too much difference from a purity level and therefore a performance level in cell culture when it comes to food grade versus pharma grade ingredients, which is also why it's pretty foolish to assume that pharma grade ingredients would be used in the manufacturing of cultivated meat. And so the takeaway here is that based on our current understanding, a lot of manufacturers as well as media suppliers are at various stages of replacing pharma grade ingredients with food, or in some cases even feed grade ingredients. And the important part is that really multiple data points and corroboratory evidence suggests that not only is this tractable with minimal technical bottlenecks, but it can happen on a very short timescale. Now, one consideration is that for things like feed grade inputs, there could be some impurity levels, some impurities within there that could pose, safety threats. And so those things have to be evaluated independently before they are used abundantly in the industry. I think what we might see over time emerge is a sort of cultivated meat grade level where you're sort of striking the balance between cost and purity and performance and safety. We're, sort of evolving a new standardization for ingredients to be used in cultivated meat production. And I think, that's something to keep track of over time. All right, so now let's talk about the two types of ingredients that are really most important to source at lower cost: the recombinant proteins and the amino acids. So anyone that has embarked on a cost analysis of cultivated meat quickly comes to the conclusion that growth factors and recombinant proteins are cost drivers. And in fact, if growth factors used in the pharmaceutical sector were used in cultivated meat production, they'd probably account for around 99% of the costs at scale. And you can see this in the analysis from my colleague of Essential 8 media, where if you look at the individual costs of each component in the media and multiply this by a factor of 20,000 to account for a 20,000 liter batch, you can quickly see that growth factors and recombinant proteins make up the vast majority of the overall cost of media. So clearly this needs to be addressed, and the question is, how are we going to address it? One of the most, sort of direct approaches is to simply reduce the overall use of growth factors in the media. And so there's various different ways that one might consider pursuing this. And just shown on this slide are going to be a couple of snapshot or representative examples where, for instance, the company 3D Biotissues has developed a supplement that can increase cell growth rates despite using growth factors at half of their normal concentrations. Others are pursuing cell engineering in ways that can eliminate entirely the need for certain growth factors like FGF2 by tricking the cell into believing that that pathway is sort of turned on all the time. And there are a variety of other ways, such as media recycling or coming up with protein engineering methods so that the growth factors stick around longer in media, for instance, that are also tractable approaches to reducing their overall use. And more of these are mentioned in a report that I published last year that you can read about and that I will be discussing in these coming slides. So one of the questions that we wanted to ask was, what would be a suitable cost profile for growth factors in the sort of cost competitive cultivated meat industry, which we set at around $10 per kilogram in this particular analysis. And the answer to that was that growth factors should probably cost at or below $100,000 per kilogram if we wanted them to, not be a cost burden for the industry. And so is that a high cost or is that a low cost? We can again look at this sort of differentiation between a pharma grade protein and a food grade protein, where pharmaceutical proteins produced in animal cells or microbial cells can often retail in the millions or up to billions of dollars per kilogram, but scaled production of proteins produced in microbial systems can, retail for as low as $10 per kilogram. And so we understand, through recombinant protein production in microbial systems that, this cost profile is very easy to obtain for basically any protein that one could imagine. And so, we talk to suppliers all the time that say that we can produce kilograms of FGF2 or IGF1 today at really low cost. But the problem is that, this is not really a technology problem, but one of markets where there's not really anyone on the other side to currently offtake that volume of growth factor. And so what we expect to sort of happen in the coming years is that growth factor costs will readily come down as production begins to scale up and that food grade downstream purification processes are implemented, which is already beginning to happen. There are various manufacturers that already sell food grade growth factors for use in cultivated meat production. And there's also some other things on the supply chain side of things that might be able to accelerate the cost for the pace of cost reduction for growth factors that we'll talk about toward the end of the presentation. Now, what this analysis also identified is that, not all of these recombinant proteins and growth factors are used equally in the media. So certain recombinant proteins such as albumin and transferrin and insulin can be used at 1,000 to over 1 million-fold higher concentration in the media than growth factors. And that, quickly means that you come to the conclusion that most of this recombinant protein volume would come from these three proteins, with actually the vast majority of it coming from a single protein, albumin, even though not all media formulations are expected to contain albumin. So what does this look like from a volume perspective? It would mean that even to supply less than 1% of global meat production through cultivated meat, we'd need to produce, millions to up to hundreds of millions of a single protein, albumin. And we'd need to produce, tens of thousands to hundreds of thousands of kilograms of proteins like insulin and transferrin, which, by the way, is about 300 to 8,000 times the current production volume for these types of proteins. And so in the short term, we think that recombinant albumin, insulin, and transferrin will be important to fill the gaps and the needs of the industry as it scales. And we know manufacturers are willing to produce these at relatively low costs today. However, we think on the longer term, we really want to avoid this scenario where we're using all of this fermentation capacity for a single media ingredient. And so we think that non-recombinant sources of these proteins are really going to be needed to supply the industry as it scales. Now, the good thing is that these proteins are really important in your cells and in your body. And so therefore, they are conserved evolutionarily. And you can find, for instance, albumin proteins abundantly in the seeds of many plants because they act as storage proteins. And it's already been demonstrated that you can take a protein isolate from a commodity crop and you can spike that into the cell culture media where it can really functionally replace the recombinant animal albumin at a fraction of the overall cost of what it would take for the recombinant version to be used. And we are funding additional research into this, and there's other groups around the world that are already working on this. And looking at the preliminary data, I think this is a pretty tractable approach, not only for albumin, but also for transferrins that can be found in plants as well as algae. Now, there's been a lot of research already into finding, sort of insulin-like compounds or molecules in crops and other organisms through diabetes research, for instance, that could be leveraged for cultivated meat. In the meantime, we've already seen Some companies like Aleph Farms partner with different companies to produce more cost-effective insulin substituents. So basically easier to manufacture versions for cultivated meat that will help to lower costs throughout the industry. And we know several other groups that are working along similar lines. Now, the last thing that I'll mention when it comes to some of these food grade media ingredients is that now that we can use food grade ingredients in media, we have a whole new sort of category of ingredients that we can explore that are already used in different foods. And so, for instance, it's already been shown that methylcellulose, which is typically used as a binder in things like plant-based meat, can enhance albumin functionality because it has sort of macromolecular crowding principles in the media that would allow you to basically bring these proteins closer to where they need to be in the cell, enable you to use fewer proteins like albumin and growth factors in the media. There's also literature from years ago that shows that it has shear protectant properties. And so, this is just one example of a ingredient that's commonly used in foods today that could have really unique functional benefits in cell culture media. And I think we'll see a lot of other creative research that explores these possibilities in the future as well. So now switching to the last category, the media ingredients that we'll talk about today, which are the amino acids. And if you look at a population of proliferating cells in the media and you track where that biomass is coming from, it's really made up from the amino acids that are being consumed by the cells during the production. Now, from a supply standpoint, a lot of amino acids, or most of them today, come from individual fermentation processes, only some of which are going to be considered sufficiently scaled for what you might need for, cultivated meat production in the future from a volume and cost perspective. And this was highlighted in David Humbert's analysis, where if you look at this macronutrients sort of cost line item here, which is the highest cost contributor in his analysis, the vast majority of these macronutrient costs are actually the amino acids. And he also highlights this sort of catch-22 scenario where amino acid costs really only become low when the cultivated meat industry is large. And so if you have a large cultivated meat industry, there's a lot of demand for amino acids. So you scale up their production, you have economies of scale, and therefore you have lower costs. But really during the early years of production, when the overall volume of cultivated meat production is low, the vast majority of the area under the curve in green here is attributable to amino acids, and that this overall high cost during these early stages of the industry would likely prevent its growth. And so how do we overcome this sort of fundamental challenge when it comes to cost discrepancies in amino acids? And one of the things that has been proposed is to source them through hydrolysates. So hydrolysates from different organisms have been used as supplements in cell culture, in pharma for really decades now. You can go to life science suppliers today and you can buy hydrolysates or peptones to spike into your media that provide a sort of source of peptides and free amino acids that can enhance your cell culture. This one happens to be derived from rice and wheat. But the question for cultivated meat really becomes, can we use these as a primary source of amino acids rather than just a supplemental source? And as described in Dave Humbert's analysis, if you take the hypothetical amino acid needs of a cell, which is shown in blue, and you compare this to the amino acid profile of soybean meal, you can see that most of the amino acids are actually, would be supplied sufficiently by feeding the cells this soybean meal. However, some of the amino acids like tyrosine shown here are not, you'd have to compensate for that by spiking in individual amino acids or by blending in another source of amino acids that contain high amounts of tyrosine. Now, if you're able to do this in cell culture, then this can have a large effect on cost. And in his analysis, he analyzed this cost of soy hydrolysates at $2 per kilo, and you can see the dramatic cost reduction in the macronutrient line item if you're able to spike in these hydrolysates and use them as amino acids. This would also potentially result in a more simplified supply chain, which you can see sort of graphically represented here, where this conventional basal medium compared to this food grade version that I was talking about earlier in the presentation, where they've added in a yeast extract, which basically cuts the number of raw materials that you'd need for the basal medium in half. This means, less middlemen in the supply chain, potentially lower costs, more simplicity, etc. So what actually is a hydrolysate? Well, you can take a sort of proteinaceous raw material, and essentially what you're doing is, through the hydrolysis process, is breaking it down into its constituent components, namely free amino acids and peptides, depending on what degree of hydrolysis you're able to achieve. Hydrolysis itself can be done through adjusting the pH, or through an enzymatic process, or even a fermentation derived process. And the sort of final product doesn't just contain peptides and amino acids, but a lot of other nutritional components like minerals or carbohydrates and even lipids can be carried over into the final product. And so you're left with this really nutritional powder that can be used in the cell culture media. So big question is, if we're going to use hydrolysates, or we think we're going to use hydrolysates in this industry, what materials should we actually pursue? And as demonstrated previously, soybean hydrolysate is a really good starting point. It has a great essential amino acid profile. That's why it's used in animal feed. That's why it's used in a lot of plant-based meat products as well. And so, soybean hydrolysate is definitely a good candidate to look at for hydrolysates. In our side streams analysis that we published last year, where we looked at potential uses of side streams of commodity crops in North America, we sort of did a similar analysis using the same amino acid requirements for a cell as modeled in Dave Humbert's analysis. And you can see that in addition to soy meal, which scored the highest in terms of meeting the cellular needs from amino acid perspective, there are also other side streams like corn distillers grains or canola meal, brewer's spent grain, as well as corn gluten meal that could have favorable amino acid profiles if they're used, as hydrolysates for cultivated meat. And so this was just sort of an initial analysis that gives you some sense of what you might want to be looking at from a nutritional perspective when pursuing different raw materials for hydrolysates. But that is really just one of many questions that need to be answered in order for us to adopt hydrolysates in the cultivated meat industry. And so in addition to the starting material that you are analyzing, you also have to look at the volume and therefore the cost of how you can actually source those today, as well as the environmental footprint, which is an important consideration since media is going to be one of the environmental impact drivers of production. This is indeed how we broke it down in our side streams analysis, looking at sort of different categories here in order to create a weighted rank for different potential raw materials. Another question is, what form should that raw material come in? So do you need it as a really concentrated isolate, or can it be more of a meal? Would it come from an existing side stream? Can we valorize other agricultural processes for use in cultivated meat? More importantly, I think there needs to be a lot of work done around the method of hydrolysase and how well that is going to work in terms of returning the type of composition that you might want for the cells, reducing lot-to-lot variability, which tends to be a challenge for these production processes, as well as looking at other unique bioactivity within the hydrolysate itself. And so it's known that certain peptides or other molecules, depending on the raw material, might have useful properties like antioxidant or antimicrobial properties in the cell culture that would be beneficial. But we also know that certain raw materials can carry anti-nutritional or even growth inhibitory factors that, if you spike in your hydrolysate, you actually end up killing your cells. And so this is often hard to sort of take apart because of the complexity and also the sort of chemically defined nature of a hydrolysate. And so these are important questions to ask, but also ones that are somewhat challenging to address. And so what we really need is deep experimental characterization done for all of the above, but also testing this out in certain cell types and in different species that are used in the cultivated meat industry. And while this is happening in the private sector to some degree today already, really everyone needs to know the answers to these questions, or else we're going to see a lot of duplicative, wasted research and efforts. And so this is really one area that I think is ripe for publishing results, protocols, and methods openly, even if you're a private company. But with that said, there's also, I think, a big opportunity for these questions to be addressed through collaborative public-private work, through some of the consortiums that have been formed already in the cultivated meat industry, since these challenges are, they're not easy to solve. There's a lot of screening, a lot of testing, a lot of work to be done. And so I think we really need collaborative efforts to sort of get us closer to the answers. A good example of a publication that I think sort of touches on some of these points, it was published last year, which is looking at a protocol for optimizing microalgae hydrolysis for use in cultivated meat. And you can see that they test out different, sort of strengths of hydrochloric acid to break down the microalgae, and they look at the resulting amino acid and glucose concentrations, as well as ultrasonic fractionation as a method to producing amino acids and glucose that could be used in production. And so this is just, this is a whole publication looking at just a really small portion or piece of the pie of this overall research question. So definitely still a lot of work to be done in order to, I think, get us to an answer of, can hydrolysates be used as the primary source of amino acids? I think the best answer to that question right now is that it's still to be determined. All right, so let's talk about now the second major step to reducing cell culture media costs, which is using less media. And this step really has to do with linking the individual cell's metabolism to the overall media formulation. And the good thing is that this work can really be done in parallel to step 1B here, because a lot of this work can go a long way to informing the future supply chain and what raw materials you might want to use in the media. So let's first start out by saying why dollars per liter is really not an appropriate long-term metric when considering media costs. And the major reason for this is that most of the cell culture media volume and weight is actually water. And therefore, measuring costs on a volumetric basis really says little about the amount of actual ingredients in the media or how efficiently those ingredients are being converted into meat. And so really the correct question on the long term to ask is, what amount in mass of media ingredients is really necessary to produce 1 kilogram of cultivated meat in a given production process? The importance of feed conversion is really highlighted in this life cycle assessment that was published last year that I worked on as well. And in this study, we assume takes place in the year 2030. So we're trying to simulate what production might look like in 2030. And we assume that some of the questions that we talked about with respect to amino acids have been resolved, and that around 3/4 of the amino acids come from hydrolysates, whereas about 1/4 of the amino acids are spiked in from individual fermentation processes. And there's 3 different media use scenarios that are included in this study that are based on data that we collected from companies and trying to really simulate what a media formulation would look like with respect to the individual metabolism or metabolic data that was received. And so what we can conclude is that, based on the data that we received, that around 600 to 800 grams of solids, so that means amino acids, sugars, and salts, would be needed to produce 1 kilogram of cultivated meat, assuming that about 70% of meat was water. But importantly that, this is based on a model, and this will need further experimental validation. Now, what's useful here is that it can serve as sort of a benchmark for the industry, because the baseline media scenario here results in a feed conversion that's nearly 3 times as efficient as chicken production. And so if you're able to really drastically improve the efficiency of meat production, then you're probably looking good in the long term with respect to comparisons on cost and environmental impact. Now, on the flip side, this data also shows us that if feed conversion is inefficient, so if we look at the high medium scenario and the amount of ingredients that are needed compared to the baseline or the low medium scenario, this would not only increase our cost dramatically, but the carbon footprint of production would increase somewhere between, let's say, 70 to 250% compared to the baseline. And that means that medium use efficiency is not only important for cost, but it's also critical to meeting, environmental or sustainability goals for the industry. So how does this compare to raw material estimates from other studies? And you can see shown here the amino acids and sugars that have been estimated across different studies that would be needed to create 1 kilogram of cultivated meat. You can see that there's some variation across studies in the amount of amino acids, as well as the amount of sugars that are predicted to be needed. With respect to amino acids, we know that if you are including a hydrolysate in your formulation, it's likely that your amino acids counts are going to be higher than if you assume that they come strictly from fermentation processes, because some of those amino acids are going to be basically an overabundance in the media. But as I discussed, we know that from a cost perspective, this is justified. For glucose, you can see that this data point is particularly high because this is actually coming from an actual cell line grown under lab conditions, what I think many would consider to be a non-optimized cell from a metabolic perspective. But a key point or takeaway from these data is that there's really a big gap right now in terms of real world data points from cells that are used in cultivated meat production. And so we need companies as well as researchers, that are growing cells at various scales of production in order to supply these sorts of data that will feed into models of the future. Now, what underlies really all of this is the cell's metabolism. And so this is really central to achieving cost and environmental goals, as we discussed. And not only would efficient metabolism lead to better feed conversion, but also less waste. And this also includes products like ammonia and a lactate that can be toxic to cells at certain concentrations and that really need to be monitored and reduced in order to have success. And in Dave Humbert's analysis, he started out with a sort of wild type metabolism where you can see produces a fairly substantial amount of ammonia as well as lactate. And then he also derives a sort of enhanced metabolism that has much significantly reduced metabolic byproducts so that you can so that productivities can be increased. Now, again, a key data gap for the industry is that these sorts of stoichiometric and mass balance equations really need to be validated based on real biomass compositional measurements from animal cells used in cultivated meat production. Until that happens and until we have those sorts of data, we're going to have to rely on models like this that are somewhat hypothetical in nature. So looking at a case study for media formulation optimization, we can see that really off-the-shelf media formulations like DMMF12, which we've talked about in previous slides, are not tailor-made. And so they often will contain ingredients that are completely unnecessary. They might be in overabundance. And this basically means that some of these ingredients, or many of them, actually become wasted in the cell culture media. And a group that has since spun out a cultivated meat company called Clever Carnivore published a paper last year where they showed that they could reduce the number of ingredients in DMMF12 from 52 to 39. And they can also simultaneously reduce the concentration of certain ingredients like amino acids and vitamins, which cumulatively can have really great effects on lowering the cost of the media. Now, this was done in the context of human pluripotent stem cells, but this company obviously is applying some of these methods and learnings into adopting these practices for cultivated meat relevant cells. But if you look into this paper, you'd find that it's quite labor intensive to do this and to screen a lot of different concentrations of all these individual ingredients. And so what a lot of people have already begun to implement are other sorts of machine learning or high dimensional designs of experiments or other algorithmic approaches to formulation optimization. I'm not going to talk too much about that in this presentation, but you can click on this paper, this review paper by Zach Cosenza at UC Davis, which provides a fairly good overview of how some of these techniques might be applied to accelerate formulation for cultivated meat. So what I think will be even more important is the concept of metabolic modeling. And we've seen in other industries using microbial cells, and more recently in the pharmaceutical industry with animal cells, that you can create what are called genome scale metabolic models that essentially map an individual cell's metabolism. And by performing these sorts of auxiliary analyses, you can identify bottlenecks in this system. And putting these things together basically allows you to really do formulation optimization in a more hypothesis-driven manner. And so the model might predict that if you were to just feed an individual amino acid in a little bit of higher concentration, then you can get great benefits in cell growth. And indeed, these sorts of simplistic solutions have already been demonstrated in different animal cells and for CHO cells in the pharmaceutical sector. And so we think that there will be really a great need to build these models and use them to inform the development of custom media formulations that we know are going to be needed throughout this industry. I won't talk too much about all of this, what's shown here, because it's worth a whole nother entire lecture. But there is a great review paper that was published by a former research fellow at GFI that I would encourage people to download and read if you're interested in starting to implement these sorts of modeling approaches, some of which we have begun to fund as well. Now, the last thing that we'll talk about that is related to what we've been discussing is recycling and valorization. And we know that ammonia is a metabolite of pretty high concern because it becomes toxic at relatively low concentrations. And therefore, there's a pretty large incentive to lower ammonia production in cultivated meat manufacturing. And there are a variety of approaches that one can already implement in order to do this, such as raising or increasing the levels of the enzyme that is responsible for utilizing ammonia in its reaction, as well as changing the media composition. So MOSA meat demonstrated that if you avoid feeding glutamine and instead feed metabolic intermediates like alpha ketoglutarate in the cell culture media, then you will limit the production of ammonia overall. And we know from techno-economic models that this can lead to increased productivity and therefore lower cost of production. Another major metabolite is lactate, and we know that this is produced in relatively high quantities, estimated sometimes around 300 kilograms per ton of cultivated meat. And a question that I think is worth asking is really how tractable is it to potentially valorize this lactate? If you're able to create a co-product, then you might have an additional revenue stream, or you could potentially offset the carbon footprint of production, since a lot of carbon flows will be going into lactate. We also know that toxicity of lactate is less of an issue compared to ammonia because it happens on about 10x higher concentration than ammonia. And so we might expect that there would be less research on how to stop its production and more research into what we actually can do with it. And I think we're hiring a research fellow that will dive deeper into some of these questions. Lastly, recycling water and other components, even including individual amino acids, might be performed in the future. And this could influence cost or environmental impact in various ways. There's also a need to, leverage these models that might be built to identify what other types of inhibitory metabolites may exist or build up over time in the media. Some of these have been identified in CHO cells, and there could be sort of parallel efforts done in cultivated meat that could also be impactful. But there's not much to really talk about with respect to these options quite yet. So the third step is really to scale the new supply chain. And once you have the lower cost ingredients, you have optimized media formulations, you just have to really scale up these ingredients, and that in turn will, by itself help to lower costs overall. But we're going to talk about some of the ways that we might take supply chain interventions to affect costs later in the presentation, since they often will also apply to equipment. So in summary, I think there is really a clear path for the steps to take, as well as some of the major R&D areas of focus to reduce the cost of cell culture media, which we've discussed. There's two major buckets. One is sourcing lower cost ingredients, which gets you much of the way there. But then the second challenge is really optimizing these formulations for efficient feed conversion. We know that there are many different pathways for sourcing lower cost ingredients, which we've discussed. The good thing is that many of these solutions can be applied throughout the entire industry. There's various tractable approaches in the literature, and that, as I mentioned, sourcing these lower cost ingredients, using food grade ingredients, et cetera, can really get you over 99% cost reduction pretty quickly compared to a pharma grade media baseline. Lastly, we think that deeply understanding metabolism and how it relates to formulation discovery and feed conversion ratio is going to be critically important, but also happen on a longer time scale. And the reason for this is because a lot of these solutions are going to need to be uniquely tailored to each cell line, and also because this research is really just beginning to happen. And so we look forward to, I think, tracking the progress in this area over the next several years. All right, so now that we've talked about the cell culture media, we can move on to the other major bucket with it, which is infrastructure. And for infrastructure, there's not as much primary data to go on because companies are really just beginning to build their first facilities. And so we're going to take an approach that's more informational to tell you a little bit about what we know when it comes to infrastructure for cultivated meat. And we're going to start with the build out requirements, as well as touch on some of the current status and scaling to meet some of the market growth estimates that have been put forth. So one of these estimates was produced by McKinsey a few years ago that suggested that by 2030, under a high growth scenario, which you can see the sort of qualifications for here, they estimated that cultivated meat could account for around 2.1 million metric tons of production. And if that were true, then it could be about a $25 billion market. Now, for context, this is about half of 1% of global meat demand, and that's specifically meat demand by the year 2030. It excludes seafood, which together with meat would be over 500 million metric tons of production by 2030. So you quickly realize that when you start beginning to talk about millions of metric tons of production, it requires a significant amount of new infrastructure, and that this infrastructure just really isn't available right now on the planet. In the same report, they estimated that to get to the 1.5 million metric ton mark, that would be about 22 times that of the current global pharmaceutical industry, which you can see represented here, where they suggest that the pharmaceutical capacity around the world is around 10 to 20 million liters, and we would need to multiply that by 22 times in order to just produce about 0.5% of the 2030 meat market demand. Another way of looking at this is from a sort of capacity gap in the amount of bioreactor capacity on Earth. And this was a sort of blog post by the company Arc Biotech, which is specializing in bioreactors for the industry. They take the midpoint of the McKinsey estimate and say that there's around 15 million liters of cell culture capacity around the world. You would need to basically 10x that in order to get 1% of cultivated meat consumption in the US, and you would need to 100x that in order to get 1% of global meat consumption met by cultivated meat. They take this a step further and they suggest that it would take about 60 billion liters of bioreactor capacity to satisfy all global meat consumption or demand. Now, this takes into account that it assumes more efficient production with their sort of hypothetical large-scale bioreactors that we'll talk about later on. And interestingly, it highlights the sort of, land requirements for such an endeavor where you could theoretically fit this amount of capacity in a land equivalent to the size of Lower Manhattan. Of course, this doesn't account for all the land that you would need to grow the media components, but nevertheless, it's an interesting it's an interesting example of some of the land advantages that could be present with cultivated meat. One of the other things that has been difficult to align on is just the scale up terminology. And so a lot of companies use different terminology when they talk about their scale of production. And sometimes this can get confusing. And so in a recent report that we are coming out with in a few weeks, we proposed this sort of guide to align on scale up terminology. Of course, this isn't a perfect fit for all different scenarios of production, and some of these numbers are dependent on many different variables that we'll talk about. But nevertheless, we think that this can be a somewhat useful guide for at least aligning on how we think about the scale of production and what amount of meat can be produced at each given scale. So where are we today with respect to scale? Based on our current knowledge, the largest known installed bioreactors are between 6 and 10,000 liters. However, there's really no confirmation that these reactors are currently operational. So they are in a facility, but they're not necessarily churning out products at this time. The largest known bioreactors that we think that are in operation are around 2,000 liters in size, and this is based off of public statements from several different companies in the sector. We're tracking at least 35 facilities around the world that have been announced or already built. The vast majority of these fit into what we would consider the pre-pilot or pilot scale of production, where you're generally creating kilograms up to dozens of kilograms per production run in these facilities, depending on the ultimate size of your largest bioreactor. There are a few facilities, such as the ones listed here, that could be considered on the lower end of an industrial facility. But again, I think most of these are under construction. They're not necessarily built and operational quite yet. Another way to look at industry growth and how we're tracking is to really estimate based on the cumulative capacity of these facilities, how much meat is expected to be produced and by what year. One of the ways that we looked at this is by surveying the industry, where we collected data from, in this case, 19 different companies that responded to this question of how much cultivated meat do you plan to produce by the end of the year 2026. We have a couple different estimates in terms of lower and upper bounds for this, but on average, the response was around 25,000 tons, which for reference is a little bit less than an average US slaughterhouse. So if we multiply this number by 5 to account for about 100 B2C companies around the world, then we can come up with a really rough estimate, a ballpark figure of around 125,000 tons of cultivated meat production by the end of 2026, equivalent to about 4 US slaughterhouses worth of production. If we assume that this turns out to be correct, then we're likely tracking toward the lower growth estimate that was put forth by McKinsey a few years back. I would like to highlight we have a disclaimer on this figure, and we talk about the limitations of this number in the report at length. And so please don't use this as a really specific number, more of a ballpark figure that we're going to have to track as time moves forward. And the key considerations to whether or not this will, become true or shall fall short or even exceed this number really depends on a couple of main factors, like the commission time of the facility. So as we mentioned, there's a difference between having the reactor capacity sitting in your facility and actual production taking place using that reactor. There's a big consideration between the cultivated meat or whether it's a hybrid product. So here we're estimating actual cultivated meat production, but we know a lot of companies are pursuing hybrid products where they're mixing in their cells with plant-based ingredients. And, if the average product had 10% cultivated meat in it, then this number could, increase by tenfold potentially. There's also, uncertainty in how successful different manufacturers will be in scaling, being able to acquire the financing that they need to build these facilities and acquire the equipment, and overall de-risking of business models for the industry that will bring in additional funds. And then finally, we're seeing challenges with regulatory in terms of just bringing a product to the market, but also the threat of bans in certain region, and so that could really influence the number of facilities that get built over time, especially in key regions around the world. All right, so now let's look at some production facility cost estimates. Starting with the Humbird analysis that looked at the cost of cultivated meat production in two hypothetical facilities, you can see that the bioreactors and the buildings cumulatively make up a fairly substantial overall portion of the cost of goods. And according to his analysis, that total capital investment for each of these respective facilities that produce around 7,000 tons of cultivated meat annually would be around $325 million or $663 million, depending on the facility that was modeled. The differences here being that facility one assumes that 20,000 liter bioreactors operate under fed batch mode, whereas facility two relies really on a 2,000 liter perfusion system, which has added costs in his model. If we look at the CE Delft study, which assumes that a 10,000 ton of annual production capacity would be met using 10,000 liter stirred tank reactors, as well as a 2,000 liter perfusion reactor for differentiation, the estimated total capital investment for this facility would be $450 million. I've summarized these two studies along with another study from UC Davis that I think is illustrative in a single table that looks at the different sizes of vessels that have been analyzed, the different production capacities in these facilities, as well as the different estimated capital investments in each respective facility. So it's important to realize that not all of these studies might be considered directly comparable because they use different assumptions that can affect the overall capital costs. But I put these all together on a single table because it gives you a sense of the expectations for CapEx costs. And one additional way that you might want to look at this roughly is by normalizing for capital cost per ton to give you a sense of the range that these different studies have shown. Now, I caution people not to overestimate this number because this doesn't mean that it literally costs $100,000 to produce a ton of cultivated meat because it's not accounting for amortization over the course or the lifespan of the facility. It's just simply a way of comparing different studies. And we can also compare these models that have been produced to some of the real world announcements by cultivated meat manufacturers. So there have been two really larger scale facilities that have been publicly announced by the more capitalized companies in the industry, Upside Foods and Believer Meats. You can see that each of these facilities are stated to have capacity for at least 10,000 tons, and the corresponding investment amounts you can see normalized here are significantly less than what's been produced in most of the modeling efforts. So I think there's two key considerations here. One is that these are just announcements. These facilities don't yet exist yet. Upside Foods has delayed the construction of their facility, whereas the Believer Meats facility is still being built right now. And we also don't know if this investment amount really corresponds to the total investment needed to reach these capacity numbers. It could be that this is an initial investment and that further investments will be needed to outfit the facility with the necessary equipment to reach these capacities. And so we're going to be tracking this over time, as well as announcements from other companies to see how well the models line up with some of the real world figures. So taking all of this together, we can generate really some initial ballpark estimates for the cost of new infrastructure in the cultivated meat industry. We might want to estimate right now, maybe conservatively by some estimates or maybe optimistically by others, that a 10,000 ton production facility would cost around 300 million. And if we wanted to reach about 0.5% of the 2030 meat market, this would require 150 of these facilities at a $45 billion capital cost. So again, just a ballpark, really rough estimate. The takeaway here is that there's high variability in the estimated costs for facilities thus far, and that also that these costs might vary significantly by region. This has yet to be incorporated into these models of the industry. So I really want to underscore that more data and studies are needed before we can have higher confidence predictions on the anticipated costs of new infrastructure. For this last section, we're going to look at some of the insights from existing studies on different bioreactor types and process considerations and how those influence the cost of production. So one observation is that nearly all of the techno-economic studies that have been performed thus far assume that the production would be taking place in stirred tank bioreactors. However, when we surveyed the industry last year, we found that while stirred tank reactors are the most predominant choice for proliferation, there actually is a diversity of approaches being pursued using other bioreactor types, as well as overall philosophies in terms of biomanufacturing. And different bioreactors can have different roles when it comes to proliferating or differentiating cells. They can have different componentry that can influence the material or utilities costs. And they can be in different sizes or shapes that can overall, influence the design footprint of your facility. And taking this all together, this means that different bioreactor types are likely to have different techno-economic outcomes. So comparing two different studies, I think, provides an illustrative example of some of the differences between bioreactors as well as influencing factors. In the Humbird study, he assumes that this 20,000 liter stirred tank reactor would be made of pharma-grade stainless steel alloy and would cost around $1.5 million. Whereas another study from researchers at UC Davis estimated that a 42,000 liter stirred tank reactor made of food-grade stainless steel would cost around $900,000, where a 211,000 liter stirred tank bioreactor made of food-grade steel would cost $2.38 million. And so you can see, I think, two different things here with this comparative data. One is that economies of scale seem to be real. And so as you get larger and larger vessels, your costs tend to decrease per unit of vessel. And so this is illustrated here in the Humbird study, where this direct cost factor really decreases substantially as you begin to scale up the size of your reactor vessel. And furthermore, the difference in material assumptions seems to also have an effect on the cost. Now, lastly, if you add in this comparison with an airlift bioreactor, which has fewer moving parts and an overall more simplistic design, an even larger vessel would presumably, according to this study, cost even less than any of these other reactors that are significantly smaller in volume. And so the type, size, and the material assumptions seem to have pretty important effects on cost. So we wanted to investigate this by asking the industry, what material are they actually using in their bioreactors? And you can see that at least some companies in the industry seem to be uptaking the use of this 304 stainless steel alloy, which is more commonly used in food production and is much more affordable than this 316 stainless steel alloy that also seems to be used in the industry. Now, one of the questions that I think remains from these data is, if 304 alloy can be used in the cultivated meat industry, why isn't everyone using it? Because it's going to be more affordable. And I think there could be some explanations, such as that most bioreactors are manufactured by pharmaceutical companies, and therefore they might be using this alloy as a sort of standard in their production. Or there could be actual technical reasons where the durability of the 304 alloy over time might not be able to withstand some of the caustic cleaning materials or other media components that are expected to be used in the industry. And so I think the adoption of this more affordable stainless steel alloy throughout the industry will be important to track as it scales up. And lastly, one other point is that other companies are exploring more affordable materials. And so, for instance, if you are able to sterilize a vessel without the need for pressurized steam, this could open the door for things like glass or other plastics to potentially be used as vessels for bioreactors. And so this will be another important, potentially important consideration to track as the industry matures. So basically every TEA will tell you that economies of scale are real and that they are an important consideration. So why is this the case? Essentially, it comes down to, as the scale of the reactor increases, your production becomes more efficient because you need fewer reactors needed for the same amount of production output. And that includes different amounts of seed train vessels and fewer interconnecting parts. You also therefore need fewer personnel in the facility, as well as utilities that are needed to operate the equipment. And finally, your cost contribution of maintenance and depreciation for that equipment decreases as well. And this is illustrated here in this TEA by ARC Biotech, which is a company that's trying to create very large bioreactors up to 1 million liters in size and really capitalize on economies of scale for production. Generally speaking, with less equipment in your facility, your total cost is going to become more dependent on the cell culture media. And again, looking at this study by UC Davis, you can see that with a 42,000 liter stir tank reactor, the facility cost contribution is above 50%. But as you get into larger and larger vessels, which require fewer overall equipment in the facility, then your facility cost contribution decreases and most of the costs become your materials, or in this case, the cell culture media that's used. So an important question, therefore, is what actual size of bioreactors can we anticipate to be used in the cultivated meat industry? And we asked companies this question, specifically in regard to planning their future facilities that would be larger scale than what they have currently. And you can see from the data that 10,000 to 50,000 liters seems to be the most predominant selection of bioreactor scale. However, it's important to note that some companies do expect or anticipate using bioreactors that are larger than 50,000 liters. Now, this number, I think, is important because essentially what we know to date from animal cell production is that really this hasn't gone larger than 25,000 liters or 20,000 liters of working volume in the biopharmaceutical industry. And therefore, if cultivated meat companies are able to start demonstrating successful production runs at volumes that are larger than 25,000 liters or significantly larger than 25,000 liters, then this could potentially be a de-risking event for the industry because it indicates that these companies would be able to capitalize on these economies of scale. However, when it comes to scale up, there's really a lot of risk that gets introduced. As we mentioned, animal cells have never really been grown at such large scales. And we know that when you get to larger scales, mixing becomes more challenging, and therefore delivery of oxygen and nutrients equally to all the cells in the vessel can become more challenging. Additionally, as you get an increased size of your vessel, there's pressure differences that get introduced to cells at the bottom versus the top of the vessel, which can have effects on viability. And so these are known technical risks, especially for animal cell culture, that are subject to things like shear stress, more so than microbial cells. Additionally, there's operational risk that takes place where if you have larger vessels, you also are generally increasing the production time because you need more doublings for those cells to reach that given volume. And you might have even more reactor surface area, especially in your largest proliferation vessel. And these things together could increase the sort of probability for contamination to take place and therefore loss of batches that would influence your profitability. And talking to different people, I think there's really different philosophies. And some people believe that we can scale up until a certain sweet spot is reached. Maybe it's 10,000 liters or 20,000 liters, depending on your product and process. And at that point, you just really copy and paste that throughout your facility. Others believe that scale up is really the most prudent path forward. And therefore, you don't stop at 20,000 liters. You keep going until you can't really go much further for whatever reason. And so I think ultimately this is going to depend on really different factors between companies, their philosophies, and when it comes to biomanufacturing, and as well as the product that they're trying to create, and therefore the sort of cost profile that they're trying to reach. But nevertheless, it will be important for future techno-economic studies to really evaluate this scale up and scale out scenario using different bioreactors at different scales, as well as different process designs or mode of operation. So we mentioned contamination before in the context of operational risk. And one of the differences between studies thus far has been some of the assumptions around how we prevent contamination in production facilities. In Humbert's study, he assumed that pharmaceutical clean rooms would be used. And he also showed that the cost of the facility would, as it increases in size, would actually outpace any of the efficiencies gained from scaling up your bioreactor. So essentially, as you increase the size of your bioreactor vessel, you also have to build a larger facility, both in the vertical and likely in the horizontal direction. And adding this sort of pharmaceutical clean room infrastructure on top of that becomes very costly. And so cultivated meat manufacturers have said that, look, we don't plan on using pharmaceutical clean rooms. We're going to grow cells in what are called clean non-classified areas or rooms with lower classifications to save on costs. However, by doing so, this potentially exposes them to higher rates of contamination because the air isn't being sort of purified or circulated at, what you would expect in a cleaner pharmaceutical room. And there's really different philosophies about this point. Some of the skeptics believe that really growing cells at large scale in production batches that take up to weeks in a non-pharmaceutical environment is really a dead end. Whereas others believe this is not really that big of a hurdle. It's just really never been done before. And so, they say, look, our bioreactor provides a sterile envelope. We could grow cells outside if we really needed to. And I think the answer to this, is that this has not been resolved yet because companies are still just entering their pilot scale production. And so this is definitely something to track as the industry matures over time. In the meantime, people are looking at other potential solutions where things like antimicrobial peptides can act as a second layer of defense if they're added into the cell culture media. And so these can prevent contaminations if something falls into the reactor, or alternatively, it can stop an existing contamination if something is detected through really sensitive sensor technology that is also being developed. So let's switch gears and talk about another important parameter in techno-economic studies and manufacturing, and that is the mode of operation. And the mode of operation basically influences how much media you're going to use and some of the densities that you might be able to achieve in your production, as well as some other factors. And when we polled the industry last year, it was determined that fed batch was the most frequent mode of operation used in the industry. However, other modes of operation are being pursued at relatively equal amounts, and basically all of the different types of mode of operation that would be available are being pursued as well. An important question to ask is, how does the mode of operation influence the cost of production? And is there a preferred mode of operation when it comes to scale-up strategy? So we know that many companies are developing these continuous or perfusion processes, but according to an analysis by Arc Biotech, these might have limitations as you increasingly scale up your bioreactor size, or if you cannot lower your media costs sufficiently. And so this is illustrated in this comparison between continuous and fed batch processes, where continuous processes shown here in orange tend to be more economical only when you have very low cost media and also at lower scales of bioreactor size or production. However, they suggest that as you increase the size of your reactor, you will predominantly run into fed batch being the more economical choice for manufacturing. And you can see the assumptions that they use for this below. And so I'm not here to tell you whether this is right or wrong, but rather to say that these types of analyses, comparing different modes of operation at different scales and under different assumptions, can be really informative for the sort of strategy over the next few years that different companies may take. And so more of these sorts of comparisons are really needed. Moving on to cell density, which is another really important cost variable or metric to track and that you'll see reported by different companies or in the literature. But what I want to illustrate here is that the context for cell density really matters and can vary based on the mode of operation as well as the type of reactor that someone is using. So just a quick illustration for stirred tank processes, for instance, as shown here, perfusion modes of operation tend to have higher cell densities because perfusion basically allows you to eliminate the metabolites that build up and limit cell growth over a certain point in time in the cell culture. And you can see that cell density shown here as it increases, you can really get dramatic decreases in the cost of production reported in dollars per pound here, assuming you're producing at a fixed scale. And so the sweet spot that a lot of companies are aiming for is, how do we increase our cell density while simultaneously decreasing our media costs? So as I mentioned before, cell density is very context dependent. And in the Humbert analysis, he suggests that perfusion would max out at 195 grams per liter in terms of cell density, because there are certain constraints imposed in his assumptions. Presumably that perfusion rate would be around 2 vessel volumes per day, which he assumes would be reasonable at the given scale of production at 20,000 liters. Now, a different report by Believer Meats in the journal Nature Food showed that they were able to generate very high densities of 360 grams per liter in sort of benchtop 2-liter vessels under a perfusion mode of operation. And so does this finding invalidate what was shown here? Well, it's important to, again, consider the constraints imposed versus the practices performed for this given study. And you can see that the perfusion rate shown in orange here is nearly 10 times as high as the rate that was assumed in this study. And so perfusion rate is really tied to the amount of media that you are going to be cycling through during your production process. And so if you have high perfusion rates, you're going to be using a lot of media. And so if these rates are not lowered, then your media costs at scale, and therefore your production costs would also be very high. And I'm sure Believer Meats knows this, but this is really just to illustrate that cell density as a single metric does not really tell you the full story. The context really matters, especially when comparing between numbers reported by different companies under different conditions. So on the last slide, I want to talk about other areas that can influence your overall cost of production that we haven't spent too much time on. And these are just other variables that can really increase the efficiency of production. So I told you before that in this CE Delft study that assumed that a facility would manufacture 10,000 metric tons of meat per year, that the capital cost would be about $450 million. But they looked at different scenarios for creating more efficient production processes, such as increasing the rate of cell doubling or the overall cell density that could be obtained, or reducing the overall media costs. And what some of these factors, the output of these factors changing means is that you'd have fewer bioreactors in the facility, as well as fewer personnel needed to operate or maintain that equipment. And so even a facility that is producing 10,000 metric tons of meat, it can go from $450 million of capital spend to $250 million. And your staff headcount can also significantly decrease. And so there are a lot of different variables really that can influence your operational costs, but also your capital costs as well. All right, so I'm going to close out with a couple of remaining thoughts and considerations, starting with the supply chain. And some of the concepts that I'll talk about can really be lectures in their own right. And so I'm going to try to keep it fairly high level. So with respect to the supply chain for things like media inputs and bioreactors and other equipment, we know that this already sort of exists in some form in other industries like the biopharmaceutical sector. But we also know that we're going to really need to reshape or repurpose these inputs and equipment to fit the needs of the cultivated meat industry. And this implies that we also will need significant investment for suppliers to go out and acquire land and build new facilities, source raw materials, and hire labor, for instance. And what we want to achieve is this sort of future scenario where you have a positive feedback loop where scaled production can lead to lower costs and better products through learning, which in turn leads to higher demand and adoption rates in the target audience, which in turn then leads to scaled production and so on and so forth. But we know that to get there, we really need a large amount of capital investment being injected into the industry. And especially within the last 2 years, as funding has really tightened up, it's become apparent that VC capital is not really the best source of funding to get us to where we need to be from here on out. And like other industries, longer term, lower cost capital that has different expectations around returns and timelines for return are really going to be critical to getting us through this sort of next phase of the industry. And we've seen in other capital intensive build outs like renewable energy that governments can play an increasingly important role by offering things like tax credits or loan guarantees, grants, or other procurement contracts. And the takeaway here is that really that none of this happens overnight, and suppliers sort of by definition sort of need to scale alongside the growth of the cultivated meat industry that they are serving. And this is inherently going to limit how fast certain costs can be reduced, because no one is going to invest the capital needed to create 100 large-scale growth factor facilities if the industry really only needs to be serviced by 5 right now. So it also might be useful to apply learnings from past work that has really focused on getting or increasing the access for vaccines and other medications in lower income countries. And this work can be sort of summarized as what's called market shaping. And some of the methods or interventions that have been used for market shaping include things like pooled procurement, where multiple companies can come together to create buyer consortiums that might be applicable for things like media or high cost equipment that is shared amongst different manufacturers. And they combine their purchasing power to give this predictable demand for suppliers so that they can go out and increase the scale of their production, but know that there's a buyer on the other side, and therefore offer lower costs to the manufacturers that are in this consortium. Coordinated ordering is similar, but relies more on sort of companies coming together to share information or joint market research, and they can come to the table with group negotiated prices and terms set out, where really the focus is on optimizing the purchasing strategy amongst companies in the industry. And then finally, variant optimization and standardization are important concepts, especially when we consider trying to take what exists for, let's say, the pharmaceutical industry and applying that to food, and some of the disconnect between specifications or standardization across those industries. And so we hear from suppliers that, they can create different pieces of equipment or consumables or inputs for the process, but they don't really know what specifications they need to hit. And so that sort of picture from a regulatory perspective will become clearer over time as the industry matures. So one of the other important concepts when it comes to long-term cost projections are learning rates. And learning rates are essentially the average decline in cost for every doubling in cumulative capacity. And the way to think about this is if you went to IKEA and purchased a piece of furniture and it then took you 3 hours to build, but then you did that the next day and it took you 20 minutes less, then perhaps on your 10th iteration, it might take you half the time to produce. And so effectively, by doing something over and over again, we get better at it and more efficient, and costs decline. And what we saw for batteries over the past few decades is that those costs declined so much that they effectively enabled electric vehicles to become competitive finally with their other sort of automobile counterparts. And so an interesting question to ask is whether learning curves for bioreactors could be analogous to what we saw for batteries. And some academics have begun to sort of retroactively analyze different renewable technology and try to be able to predict whether or not something is going to be on a robust learning curve or not. And the axes at which they have analyzed this essentially come down to design complexity and need for customization. And so from a bioreactor equipment perspective, where we're taking something that's pretty complex and maybe has too many bells and whistles attached that we have for pharmaceuticals, maybe we can move that to a lower design complexity space, something that's perhaps more modular in design, as well as lower the need for customization in order to ensure that the cost of bioreactors is significantly less than what they might otherwise be today. So you'll often see many people try to compare cultivated meat to the struggles of the biofuels industry around 10 or 15 years ago. But I think people are missing an important consideration, which is that cultivated meat really does not need to directly compete with commodity forms of conventional meat from the get-go. And so people really are not willing to pay $30 for a bio-based fuel per gallon if they can buy one at $3 per gallon. But the same is not really true for meat. People are willing to pay much more premium prices for different cuts of meat or from different species, et cetera. And this market is not necessarily small. It's billions of tons annually in terms of production volume. And therefore, I think cultivated meat might analogize better to the solar panel industry, where initially the production of solar panels was way too high. And the only industry or buyer that was willing to work with this cost profile was the satellite industry in space. But this actually enabled solar panel manufacturers to start learning by doing and get better at manufacturing solar panels and thus lower their costs, eventually bringing them to consumers on Earth. And then over the past few decades, we've actually seen such precipitous cost drops that this has become the most cost-efficient form of energy production that humanity has really ever seen before. And so an important point is that cultivated meat will probably require at least 13 to 15 doublings from its current capacity points just to fulfill the demand of this premium and specialty meat market. And so there's plenty of room for learning rates to emerge in the cultivated meat industry before it necessarily needs to compete with lower cost items like chicken, pork, or ground beef. Finally, another consideration is that cultivated meats companies don't need to limit themselves to just selling meat. They're doing a lot of research and development on things that can be used by other cultivated meat manufacturers or even in other industries. And so that includes things like media formulations or other media inputs, cell lines, or other process and equipment related IP. And we've begun to see different cultivated meat manufacturers partner or license their technology to suppliers to manufacture, let's say, media or other inputs for sale to other companies in the industry, thus generating a revenue stream for those respective companies. And the same is true for equipment, where we've seen companies like Stakeholder Foods sign deals with a plant-based meat manufacturer to leverage its 3D printing technology and inks. And lastly, it's possible that other co-products could be valorized to provide meaningful revenue streams for companies. We mentioned lactate earlier on, so that's one option that should be explored, but also other things could be potentially valorized. So with all of that said, where do we stand? So in this figure, I've compiled various data points from scenarios in different TEA studies, many of which we've talked about throughout this lecture. And I've organized this by the scale of the reactor that's used in the models. And you can see from these data points that there's not really any clear trend here, although there is some indication that scale does matter, as we discussed. However, you'll notice that there is a wide range of values that have come out of these studies in terms of the predicted cost of cultivated meat at various different scales. And I think importantly, contrary to some opinions, there are actually multiple studies that suggest cost competitiveness could be achieved. It's just that many of these scenarios assume very large scales of production or have pretty favorable assumptions that we wouldn't necessarily expect to occur within the next few years. I think this also underscores a need for more TEAs to get a more precise value that comes out of an accumulation of sort of evidence. In particular, I think there's gaps in existing TEAs with respect to other bioreactor types that might be used in the industry, as well as accounting for the influence of mass gain during differentiation, as well as the need to analyze the cost of hybrid products that we believe will be predominant in this industry throughout this decade. It's also important to realize that many of these studies don't include all of the production steps, and so they stop at the cell harvest and sort of don't account for any costs that might occur after that. And there's also a need to analyze the profitability and the payback time for different facilities or different scenarios of production, especially as companies need to acquire large amounts of funding to build these future facilities and overall de-risk the business proposition for investors. So generally speaking, the existing cost models suggest that price parity, especially in the short term, is not really predicted to be achievable. But some cultivated meat companies are saying otherwise. So for instance, the CEO of Prolific Machines suggests that a less than $7 per kilogram cost for beef production could be reached in their first commercial facility, with costs being reduced further in future facilities. And the CEO of O-Meat saying that price parity could be achieved today for their beef product in 10,000 liter bioreactors that they plan to have in their pilot plant. And so since these sort of predictions are on a relatively short timescale, then we should be able to validate or invalidate them in the next few years. And if these predictions do end up holding true, then it would suggest that some of the things in our existing cost models might be missing some important details. Another data point that I've been tracking is really claims about profitability. And you tend to see profitability mentioned really in the context of more high-priced or premium products, like the ones that I was referring to in the context of solar energy. And so, for instance, the CEO of Umami Bioworks has said that they expect profitable production for products like eel and grouper at 2,000 liter reactors, which we know is the scale at which some companies are operating today, and that large profit margins could potentially be obtainable if companies are able to reach very large scales of production for premium products like tuna. And so collectively, I think these data points are interesting to sort of hold on to and see how they track over time. Now, with that said, I think putting that all together, I don't think we actually know the cost of cultivated meat production today or in the future per se. There are a lot of different production scenarios and approaches being pursued. And so a single number really is hard to provide. And as I mentioned, I think there's a need for additional studies that are going to enable us to get a higher degree of confidence in the expected range, because right now assumptions tend to be, all over the place, and that results in numbers that are somewhat all over the place as well. And so as we learn more about the practices in the industry, I think that range will narrow over time. I think it's also critical to provide context for the product and whether or not it's a hybrid, especially when discussing how costs compare to conventional meat, as well as overall viability. And so the viability of the technology as a whole looks a lot different if you're talking about 100% cultivated chicken versus a hybrid of a premium product, for instance. So I think we could say with some degree of confidence at this stage that if you're pursuing this sort of higher end premium product, then a pathway where you're using 100% cultivated animal cells could be a tractable approach to achieving cost competitiveness. Whereas if you're pursuing commodity meats, which I define as things like chicken, pork, beef, and certain seafoods, then a hybrid product at lower inclusion rates of animal cells is really going to be the most tractable way right now to approach cost competitiveness. But it's worth mentioning that I think this timeline is uncertain. The scale at which this happens is uncertain. And success is going to really ultimately depend on many of the variables that were discussed in this presentation. So hopefully you've learned something about cost drivers of cultivated meat in this lecture. You may have noticed that I presented data from an industry survey that hasn't really been publicly released before. And so you can join us next week for a webinar for an overview of this new report that we're coming out with called Trends in Cultivated Meat Scale Up in Bioprocessing, which I think provides a lot of insights into the different practices that companies are pursuing in the industry. Thank you for listening. All right, so we'll get to some Q&A now.
Hi, thank you so much, Elliot. That was a great presentation. Really appreciate it. And as a reminder, please add your Q&A to the Q&A box, and let's hope that we can get to it. We have a lot of questions, so let's get started. Elliot, this first question, which plant-derived albumins have been looked at so far? Are any of them better than others?
Yeah, it's a good question. So when I was talking about looking at albumins from plants, so far there's been one publication from a group at Tufts University. So you can check that out. It's linked in the slides. And they particularly found that rapeseed plant isolate perform the best in their hands and in their cell types. As I mentioned, we're funding other research along these lines. And I can say that a lot of legume crops are being screened for their performance. But a lot of these data are not yet published. So I think you can expect to see more of this sort of information coming out over the next year or so. And I think what will also be important is to, validate whether these sort of isolates alone are sufficient, or that other things need to be added in as sort of auxiliary functions of albumin, which is a very complex protein in the media. So that's something to look out for.
Thank you. The next is two questions that are related to each other, so I'm going to read them both. Do you have any clues on the benefit of using recombinant proteins produced in plants versus fermentation? Obviously, it eliminates the use of bioreactors, but purification is then a challenge. And this person is asking mostly about the environmental impact. And as a related question, there are some companies that are developing crops like tobacco, genetically modified to produce growth factors. Was there any consideration of this technology in your review? And if so, what is your personal and scientific opinion on it?
Yeah, so these are good questions. This refers to what many are calling plant molecular farming, which is basically just recombinant protein expression using plants as a host rather than microbes. So I think, in terms of technique or host, there's pros and cons to each. So plant molecular farming will probably have, a lower capex because you don't need a facility to house bioreactors, for instance. They might be some sustainability benefits to using plants, which have, pretty low input requirements. But they also can take a very long time to grow. And so, microbes double every couple of hours, whereas plants, might take months to go from seed to sow. And so there's pros and cons of each. I think it would be valuable for there to be head-to-head comparisons done with respect to, these sorts of, putting these pros and cons in a matrix against different recombinant protein expression technologies. And yes, I do talk about this in a report that we published last year that's linked in these slides that talk about growth factors. So I would encourage you to check that out.
Great. Next question. Previously, there has been some discussion about minimizing or completely removing the dependence of using antibiotics in complete media compositions. How do you see this aligning with the goals to use food-grade products? Is there a concern with the potential for greater contamination? And as a related question, should we expect antibiotic use with large bioreactors?
Yeah, so these questions have to do with antibiotic use. I think one thing I'd like to clarify is that if you're using a cruder input for the media, that's not necessarily really probably going to affect too much around contamination, because regardless, the media components get sterilized before they enter the bioreactor anyway. So I think that's an important thing to note. Regarding antibiotic use with larger reactors, I think antibiotic use, there's a lot of other reasons why you don't want to use antibiotics, not just cost, but also their general effects on cells if you're using them at concentrations that you need to actually be effective in cell culture media. And so we have a paper on this and a blog post by my colleague Claire and one of our research fellows that goes into talking about this. But I think there's really strong incentives not to use antibiotics, and so we don't expect them to be used. But as I mentioned, I think there will be pursuit of other sorts of auxiliary second layer of defense type of methods, such as using antimicrobial peptides, which can be potentially just food safe, and so you might not have to worry about them from a safety perspective or from interfering with the performance of your cells in the media. So I think that's something to track. And I see Claire is dropping some of these links in the chat. So thank you, Claire.
Okay. Another question, which is simple, but well, elegant maybe. Why have the pharma grade ingredients persisted as long as they have?
Yeah, so I think one thing is that it takes time to switch. There is still some sort of validation work that has to go on. But I think more importantly, it's, a lot of people, they get their information from what's published in the literature. And the industry is several years ahead from an R&D perspective from what you'd see in the peer-reviewed literature. I mean, these companies started with, in some cases, hundreds of millions of dollars, 5 to 10 years ago. And research labs have just started a couple of years ago on a much lower budget. And so, there's a lag between what's available in research labs and coming out of academic institutes versus the practices being in place in industry.
Okay, thank you. Next question. How do you see the possibility of using animal byproducts such as feathers, blood, and offal as sources of nutrients for cell culture media formulation? In a circularity perspective, and considering that the animal industry won't be extinct in the coming decades, shouldn't we be addressing the possibility of using animal byproducts as raw materials for the transition to a totally animal-free industry?
Yeah, interesting question. So there are certainly academic groups looking at this. I know been some papers that have been published on this, taking hydrolysates of blood or bone and things like that, and using those as media supplements. So I think sort of regardless, to some extent, that is going to be pursued by some people. I think, however, the philosophical underpinnings of the industry aren't really aligned with the use of animal products. And so I think, personally, it's probably best if efforts are spent primarily on making this technology work without the use of animal products, which, to our knowledge, should be entirely possible and feasible.
Okay. Are there any good papers reviewing the media costs per tier of bioreactor and scale? Just to show what the land cost is for that lower Manhattan bioreactor capacity as an example, but at different scales.
Okay. I'm not sure exactly what this question's asking. I think in general, the media cost, as I mentioned, it's going to vary based on the cost of the ingredients, but also how it's used in the process. And I think there are papers that sort of touch on each of those aspects to some extent, but in general, I think we need a lot more of the latter, which is looking at media and how it's used in different processes under different modes of operation. So I don't have anything particular that I would share to that, but there are some publications that I touched on in the presentation that are worth reading through.
Okay. Next question. Why are airlift bioreactors estimated to be cheaper than stirred tank bioreactors?
Yeah, so I think it's not like I'm not an expert in this per se, but I think basically it comes down to simplicity of design. And so airlift bioreactors don't, they have a different method of agitation or mixing. And so there's no impeller, there's no moving parts really within the vessel. And that just makes it simpler from a materials and construction perspective.
Okay. What is the industry's view on genetically engineered cell lines that try to address some of the issues you discussed, such as engineered cells that can grow in suspension to maximize density in bioreactors?
Yeah, good question. So I explicitly mentioned at the first slide that I didn't incorporate so many cell line considerations into this talk, but you are correct, whoever asked this, that genetic engineering and those sorts of tools can address, to some extent, a lot of the issues that we talked about. And so the industry stance, well, we surveyed the industry and we have a report on that in our cell lines survey. So I will make a note to drop that in the chat. Maybe Claire or my colleagues can post that in the chat while I'm talking. But essentially, different companies have different views, and it really depends really on where they sit geographically or where they intend to market their products. So you'll see companies in Europe tend to be non-GM because if they wanted to use genetic modification, then they'd be in for quite a long haul when it comes to regulatory process. But companies that want to market in the US, in fact, Upside Foods' product does incorporate a genetically engineered cell line. And so that's already been approved and it seems to be more permissible. And so I think you will see more and more genetic engineering or editing tools become incorporated into the cells to address some of these concerns because it is probably one of the best tools available to address some of these problems.
Okay, I see that thank you, Claire, for dropping the link in for the cell line survey. Next question. Do any of the reports discussed detail the energy and water consumption that bioreactor facilities require?
Yes, so when you're doing these sorts of analyses, a lot of when you do equipment, sort of calculations and that sort of thing, a lot of this auxiliary information gets attached. And so I would encourage you to look at, whoever asked that, the supplementary information from our life cycle assessment. There's a table that goes into power consumption and other sorts of estimates for these things. But I will say that for sure, more of that information, becoming available in these sorts of studies would really be useful for the field.
Okay. When looking at the cost of perfusion by looking at the volumes of media required, would this volume be as concentrated as media for fed batch, or would the concentrations be lower and therefore costs not necessarily higher?
Yeah, so I think basically what this is asking is generally, in fed batch cultures, what you might do is for ingredients or inputs that are consumed in high amounts, you might just spike those in individually in high concentrations. And so whether or not that happens with perfusion, or the extent of that happens with perfusion, I'm not necessarily sure of, and it might vary depending on exactly what you're doing. But yes, this part in general, this sort of, you might need to spike in certain components. That sort of can make volume based calculations somewhat hard to interpret. So I would agree with that statement.
Okay, great. Next question. As bioreactors scale to 10,000 liters and larger, what can be done to maintain O2 levels while preventing cell stress? Drill pipe spargers seem to be limited vis-a-vis dissolved O2 rates at scale. I've heard of porous spargers used in the pharma space. What are your thoughts on the viability of porous spargers for commercial scale mass transfer?
So I think this is a good, interesting question. A little bit outside of my exact area of expertise and area that we focused on, but that I focused on thus far. But I think, with some of my colleagues, we'll be looking into some of these scale up challenges a little bit more as time progresses. One of the things that I think is interesting that I have, just mentioned, but I have no idea about tractability, is the use of nanobubbles for oxygenation. Could be some interesting technology that could address potentially oxygenation at very large scales. But yeah, we'll be looking into some of these scale up challenges more as time moves forward.
Okay, great. Next question. You mentioned a technical risk with large bioreactors associated with taller reactors, leading to higher pressure for cells on the bottom, leading to issues with viability. Do you have any references or literature to that we could read describing this phenomena?
I don't know if I have any references off the top of my head. My understanding of this question, again, is a little bit outside of my direct area of expertise and focus. But from what I understand, this is not necessarily about, cells getting squished to death at the bottom of a reactor, but more that solubility of certain ingredients or oxygen can, differ with different pressure. And so I think those sorts of things together can cause issues that lead to viability. Things like accumulating cells at the top of the vessel, depending on the cell type that you're growing as well, can be issues. So yeah, again, these sorts of questions on technical risk with scale up is an area that we'll, begin spending a lot more time on at GFI.
Looking forward to that. Where do you expect most production to take place in the future? The individual companies producing their own products, or through cultivated meat CDMOs?
Yeah, this is an interesting question. We gathered some data on this in the bioprocess survey that we'll be coming out with in a couple of weeks. Obviously, I don't know the answer. All I can say is that there's a need for CDMOs to sort of, let's say, remove the capital requirements that it takes to build a facility for these startup companies, especially in today's funding environment. And I think some of these processes will be relatively transferable. And so if you have a process that you're designing, why not manufacture it in another country through through a CDMO to increase the scale of your production? sooner rather than later. So I think at least in the short, medium term, within 5-7 years, we'll see CDMOs becoming much more prevalent. Whether that holds true in the longer term, I'm not quite sure.
Okay. Next question. In current food production, lines are sometimes mixed with different products to achieve higher efficiency and returns, but this means cross-contamination for some allergens. Do you think cultivated meat can reduce this issue? Current gluten-free products, for instance, are far more expensive than their regular alternatives.
All right, so these are the questions I'm seeing for the first time, so I haven't had time to think about them. But I think with respect to allergenicity, there will be controls in place that you implement sort of across the food industry, especially with respect to inputs. You can't do much about controlling allergens that are inherent to the cells themselves unless you genetically modify them away potentially in the future. I think we'll see in facilities that some companies will use the same equipment to manufacture different products. So products that might contain allergens versus others that don't. So that's a consideration that these companies will have to take into place. And I guess cell lines in general, I mean, they're very easy to store and track. And so that makes traceability potentially a lot easier in the cultivated meat industry than conventional meat. So I guess those are some considerations. I'm not sure if I answered that question fully, though.
Okay, let's move on to the next one. Has there been any work on synthetic peptides as alternatives to growth factors and albumin?
Yeah, so there have been. This is definitely an area that we get asked about a lot from researchers, especially with respect to all the tools available now for making proteins efficiently or designing them in silico. So I think some areas that you might look into is creating variants of growth factors that might be more thermostable, so that they don't degrade easily in the cell culture as readily. You could have them have multiple binding domains, so you're combining multiple growth factors in a single protein. Lots of different, I think, possibilities there. So yes, I think there is work on that going on. I think the question is, from a regulatory standpoint, there's going to be scrutinized a bit higher than using a sort of a protein that's found already in the genomes of animals, but certainly not something that I don't think you could overcome. And if you are designing something that's more thermostable, then you also have to look at it from a residue perspective, which regulators also look at. They scrutinize that quite a bit. And so if you're banking on something being degraded during cooking, but you have a very high thermostable protein, that's a consideration you might want to look at downstream.
Okay, we have a few more to get through before our time ends. In your opinion, what is your perspective on the current state of microcarriers for augmenting cell expansion capabilities in microreactors? And what type do you think are the most promising? For instance, there's a new generation that dissolves automatically, which allows to save on enzymatic harvesting of cells. But are there additional ones more promising on the horizon?
So we know from our survey data that microcarriers are being used in the industry by various different companies. I think there are different philosophies on this. Generally speaking, it's hard to get the densities that you can achieve with single cell suspension in using a microcarrier. I think making a microcarrier out of an edible substance or something that's biodegradable is sort of a starting point from which to work on or advance from what's been done in other industries. I don't have any strong opinions about microcarriers, scaffolding technologies in general. I think there's a lot. I still call it the Wild West because there's a lot of materials to explore and a lot of methods of fabrication. So something interesting that I think we will, be watching out for to see, especially from a cost perspective, can microcarriers get you there compared to other production methods?
The next question is, I think, stems from your market shaping portion of your presentation. What industry consortia are currently doing pooled procurement or coordinated ordering?
Great question. I think the answer is none right now, but it's something that we would love to see happen, and some people are certainly thinking about this. So if you're interested in this in particular, let us know, and we can maybe put you in contact with people that are exploring this idea.
Okay. Another interesting factor that should drive cost is the shelf life time. Due to the sterile fabrication, cultivated meat promises longer shelf life at retail. How do you see this impacting the costs or assisting with commercialization?
Um, yeah, I mean, it's interesting to think about. I'm not sure how much cost factors into shelf life. Certainly, it could help with food waste, which we know is a huge problem in meat and seafood in particular. So yes, I think shelf life is something that's been brought up over time where, theoretically you have a sterile process. Once those cells come out of the bioreactor, however, they are going to be exposed to air, so there will probably be some inoculation, but certainly at lower than what you find in a conventional meat product. So I think this is something that we're waiting to see the data on. Show the data on shelf life stability. So in some of the regulatory documents, I think in good meats in particular, they have some sort of long-term stability that they looked at. It's not necessarily shelf life, but I think after freezing. So yeah, it's an interesting area to explore. It could cut down on refrigeration and cold transport costs. A lot of different things that are sort of interconnected from a supply standpoint could benefit from these, theoretically longer shelf life products at room temperature. So yeah, show us the data. That'd be great.
Okay. One last question, maybe the most important question. In light of the recent funding challenges and layoffs in the industry, what advice do you have for early career researchers and recent graduates on effectively building a career in cultured meat?
Yeah, great question. I think one of the things that I've always said to students is that, I mean, you don't want to get too antsy. Like I think people really feel this urgency, like fear of missing out on this industry. It's just going to pass them by while they're, training or doing a PhD program or something like that. But I think that the answer to that is like this is a very long-term game. This industry is just starting and will be around a lot longer than it will take for you to, do your undergraduate studies or your PhD. So I think, building the skills in some of the things that we talked about today, whether it be biology or biochemistry, chemical engineering, mechanical engineering. I mean, there's so many auxiliary fields that sort of intersect with cultivated meat that training for in one of those can pay dividends down the road from a career perspective. And yeah, I think you don't have to work at a cultivated meat company to contribute to the industry. I think that's another important consideration. Most of the companies, probably by number, will eventually be suppliers or other sort of auxiliary companies along the value chain. And so I think you want to keep an eye out for how your skill sets might contribute to those companies' efforts as well. I think with that, we can wrap up exactly at the 2 hour mark. Thank you everyone for attending. I hope everyone learned something today. But yeah, as mentioned, we'll share everything afterwards. And yeah, please send me your feedback if I was wrong about anything or right. Thank you.